Hello and welcome back. Today we're going to be looking at one more important concept in biochemistry, particularly metabolism, which goes by by substrate level phosphorylation. In one of my earlier videos, you must have heard of uh, me speaking about electron transport chain. In most cells which have mitochondria, electron transport chain is the most important mechanism by which cellular potential is harnessed to synthesize ATP. But I also mentioned that that's not the only mechanism by which cells can synthesize ATP. Uh, there are mechanisms which are independent of mitochondrial involvement where ATP is still synthesized, such that the ADP-ATP ratios can be kept optimally in favor of ATP for cell survival. Uh, and these mechanisms often are categorized under the term substrate level phosphorylation. Think about RBCs, mature RBCs, mature RBCs that circulate in the blood carrying oxygen bound to hemoglobin would have expelled most of their internal organelles. Much of the space within the RBC is exclusively meant for storing hemoglobin, and they practically are full of hemoglobin, and their only job is to carry dissolved gases. Okay, they rely on substrate level phosphorylation. Substrate level phosphorylation can be simply understood by realizing the fact that in not many pathways, ATP is synthesized directly by transfer of a phosphoryl group from a high energy compound to ADP. You simply take a high energy compound and use it as a source of phosphoryl group and transfer the phosphoryl group to ADP, and you will be able to synthesize ATP back again. And in order to understand substrate level phosphorylation, you got to keep in mind a few things. One thing is you need to know what truly are high energy compounds. The other thing you need to know is the energy that is required for ATP synthesis. Third thing you got to keep in mind is, although you have involvement of high energy compounds, they're still going to be dependent on enzymes to catalyze reactions where high energy compounds are used up as source of phosphoryl group, where the phosphoryl group is transferred to ADP. Okay? So these enzymes generally are the ones that will bind the high energy compound as well as ADP, and they catalyze this reaction of direct transfer of phosphoryl group from high energy compounds to ADP such that ATP is synthesized. What are high energy compounds? High energy compounds are those compounds that have energy of hydrolysis greater than minus 25 kilojoules per mole. Minus 25 kilojoules per mole. These compounds are enzymatically hydrolyzed, and those reactions where the compounds are enzymatically hydrolyzed are characterized by a change in Gibbs free energy of greater than minus 25 kilojoules per mole. So delta G naught prime will be greater than minus 20 kilojoules per mole. And whenever you have such compounds operating in an enzyme catalyzed reaction, those compounds are referred to as high energy compounds. That's something you got to keep in mind. But particularly when it comes to substrate level phosphorylation, you also need to know that high energy compounds must be able to yield free energy change of hydrolysis, which compensates the energy requirement for ATP synthesis. And if you're looking at ATP synthesis under cellular conditions, ATP synthesis itself, okay, the energy required for ATP synthesis is about 31 kilojoules per mole. Okay, so you need to work with high energy compounds which have free energy change of hydrolysis greater than minus 31 kilojoules per mole. It's as simple as that, okay? And you also have to understand that substrate level phosphorylations, although directly involve transfer of phosphoryl groups from high energy compounds to ADP, they still have to be catalyzed by an enzyme. That's very, very important, okay? These three considerations you've got to keep in mind to be able to understand substrate level phosphorylation. We'll look at one example and you can apply the same logic to be able to understand a couple of more substrate level phosphorylation events. Okay, the example we are looking at is that of phosphoryl pyruvate serving as a source of inorganic phosphate for synthesis of ATP in a reaction that is catalyzed by the enzyme pyruvate kinase. Okay. Before we go there, just have a look at the ATP structure. Okay. Forget about the top entry. Look at the middle entry. This is the ATP, which is complex to magnesium, which is what is going to be the case in most conditions under the cellular dynamics. ATP has got a free energy change of hydrolysis of about 31 kilojoules per mole, which simply means that when you hydrolyze the terminal phosphoryl group, okay, and remove it as inorganic phosphate, leaving behind an inorganic phosphate and ADP, okay, leaving behind inorganic phosphate and an ADP molecule. Such hydrolytic reactions are characterized by a delta G value of 31 kilojoules per mole roughly, okay. ADP, by the way, is also a high energy compound. It can hydrolyze, it can exhibit hydrolysis of this phosphate, resulting in formation of inorganic phosphate and AMP. And these reactions are also high energy reactions, but AMP alone is not a high energy compound. Okay, that's something you got to keep in mind. Okay, so what we'll do is we'll straight away move on to the case of phosphoryl pyruvate and look at it as a high energy compound which facilitates synthesis of ATP by substrate level phosphorylation. Okay. Now, what you're looking at here is phosphoryl pyruvate. The structure is self-explanatory. The pyruvate, which is CH3, C double bond O, COO minus. This is pyruvate, CH3, C double bond O, CO minus, is in the enol form. So it's phosphoenol pyruvate. Okay, it's just that it's in the enol form. So you have CH2 double bond C, and this carbon atom is internally attached to an inorganic, attached to a phosphoryl group here, and then you have the carboxyl group. Collectively, the hydrolysis of phosphoryl pyruvate to inorganic phosphate, which is depicted here, and pyruvate has got a free energy change of minus 62.2 kilojoules per mole. Okay, kilojoules per mole, and it comes from two separate events. One is hydrolysis of phosphoryl pyruvate. This bond is going to be hydrolyzed, okay, and you get inorganic phosphate and the pyruvate, but in the enol form. Okay, so this is the enol form of pyruvate. Now this tautomerizes to keto form of pyruvate. Is simply the double bond goes here, and it picks up, this carbon picks up a proton, and the lone pair of electrons from oxygen come here. And what you get is CH3C double bond O, CO minus, and that's the keto form. It's more stable. Okay, unstable form is the enol form, and unstable form will have higher energy, and stable form will have lower energy, and the net difference is seen as this delta G value. So the hydrolysis of phosphoryl pyruvate to pyruvate has got a very, very large delta G value, which is attributable to hydrolysis as well as tautomerization, collectively accounting for minus 62.2 kilojoules per mole. And that means that when an enzyme has access to phosphoryl pyruvate for a catalyzed reaction, where the hydrolysis is the reaction that is taking place, there is energy yield that can be used for something else, and cells don't let it go waste. What they do is they use such energies, especially when a phosphoryl group is coming out as one of the products. They're going to use this energy as well as the product of the reaction, which is phosphoryl group, to transfer this phosphoryl group to ADP. Okay, so this phosphoryl group will be transferred to ADP such that you get ATP. Okay, and this is within the thermodynamic and molecular design of the enzyme itself. Nature has designed it in a way that when an enzyme catalyzed reaction experiences an yield of energy which is far greater than what is required for synthesizing yet another molecule, it may be coupled, okay, for synthesis of a molecule which is desirable. In our case, it's ATP, okay. You want to be able to synthesize as many ATPs as possible. So phosphoryl pyruvate is going to be used up by the enzyme pyruvate kinase, and it's going to hydrolyze it to pyruvate, which will entail both hydrolysis of the phosphoryl group as well as tautomerization of tautomerization of the inner form to the keto form, such that you get an energy output of minus 62.2 kilojoules, which can be used for synthesizing ATP. Okay, so ATP requires only half of this energy to be synthesized, and that's what happens. So as the enzyme hydrolyzes phosphoryl pyruvate to pyruvate, and an inorganic phosphate, which is mentioned here, ADP is going to be bound to the active site as well. So actually, it's the ADP that initiates the attack, but let's not worry about that. The net outcome is the phosphoryl group will be used up for phosphorylating ADP to ATP, and that's how you synthesize ATP from ADP in a reaction which is energetically driven by high energy compound. Okay, high energy compounds are those that have free energy of hydrolysis of greater than minus 25 kilojoules per mole. But in order to be able to participate in synthesis of ATP, the high energy intermediates need to have a phosphoryl group or must be involved with something which will eventually have a phosphoryl group donor. But in most cases, it will itself have a phosphoryl group, and it must have a delta G value for that reaction, which is greater than what is required for synthesizing ATP, which is about 31 kilojoules per mole. So when you look at these two energies, the net outcome of these two energies balancing out is that when phosphoryl pyruvate is acted upon by pyruvate kinase for synthesis, for conversion of phosphoryl pyruvate to pyruvate and conversion of ADP to ATP, the net energy outcome is minus 31 kilojoules per mole or 32 kilojoules per mole. These are under standard conditions. Okay, it will be slightly different under cellular conditions, but still the delta G values will be fairly negative, such that the flux is maintained towards synthesis of pyruvate and ATP, and that's how cells operate through glycolytic pathway at this step to be able to convert phosphoryl pyruvate to pyruvate with concomitant phosphorylation of ADP to ATP. I hope this video has given you insights into how energy compounds are used up by enzymes that catalyze phosphoryl group transfer reactions, typically what is referred to as substrate level phosphorylation. And you can apply the same logic to be able to understand how two more molecules, 1,3-bisphosphoglycerate and phosphocreatine, also serve as substrates for enzyme catalyzed reactions involving substrate level phosphorylation. I hope you like this video, and if you liked it, please subscribe to my channel. Thank you very much.